next presentation would be given by Jerry Glickson, effects of Lonidamine on intra-extracellular pH and bioenergetics of human melanoma xenografts and on the response to systemic chemotherapy. It's a long title, enough to give you time for this.
Well, first of all, I want to thank the organizing committee and particularly Gil for inviting me here. Really, I love coming back to Israel and seeing a lot of family and seeing a lot of friends. Gil actually got me started in vivo NMR of tumors in an indirect way. We were at a cancer center in Birmingham, Alabama, and we always were promising to do cancer research, and we were just studying high-resolution NMR of various biomolecules related perhaps to cancer. And finally, we got down to let's look at some real cancer cells, and we came across this paper by Navon and a lot of intermediate authors, and then Bob Shulman. And we said, and they were looking at Ehrlichiocytes cells in NMR tubes. And that's what we started looking at. Now, there were problems. These cells would settle out, and they were very high density, and they were dying as we were looking at them. And when we gave them cancer drugs, they died a little faster. You know, it wasn't quite reality, so that got us going into looking at real in vivo tissue, which was well perfused, tumors which were generally much better perfused, and we got some very useful results. But Gil was the stimulus for doing this. And as you'll see, I trip across Bill again in another problem that we've been studying. So he keeps on popping up. As you get new ideas, you find out Gil's been in it already. So I'm going to tell you about melanoma. As you know, it's a nasty disease. And it's only about 3 or 4% of skin cancers, but you got to remember, skin cancers are probably the most common form of cancer overall. 80% of skin cancer deaths are from melanoma. And the rate of increase has been extraordinary, particularly among Caucasian populations. So it's about 4% a year, and it's scheduled to overtake lung, breast, and prostate by around mid-century. It's the 6th most common cancer in the U.S. 80% are cured with surgery, but if the disease recurs with metastasis, there is no really good way of treating it. So drug targeting has had some promise recently when they found that about 50% of human melanomas carry this mutation in BRAF, and they could target drugs specifically to this mutant protein, the kinase. But the tumors disappeared almost miraculously, and but with dismal results in the end. They always come back. So we have a different approach to dealing with this. We had been working, actually it was Dennis Leeper at Tom Jefferson University, one of our collaborators. He had a program project since the early 90s to develop methods to selectively acidify melanoma. Dennis's main interest is hypothermia. I joined their effort around 1998, and my interest was more in chemotherapy. So I've slanted that in that direction, and we found that we could, in fact, selectively acidify tumors. I'll explain the whole scheme for doing this. In fact, why don't I just show it to you right off the bat? Okay, this whole Bubba mice that goes first. So these are drugs. The main scheme is this. We know that cancer has a metabolic proclivity or preference for glucose. as its primary substrate. And we figured, well, why don't we just take advantage of that? And the glucose comes in through the glucose transporters and goes through the glycolytic chain to pyruvate, and from there to lactic acid. So we said, okay, you like glucose, we'll make you choke on glucose. So we increased the glucose concentration to around 26 millimolar in the blood in order to maximize the amount of lactic acid that is being produced. And furthermore, we figured that some of the pyruvate would go into the mitochondria and go into the TCA cycles, so we shut down this pathway by giving MIBG, which is an inhibitor of site 1 and oxidative phosphorylation. So now all the flux is going into glucose. So the cell now is confronted with a lot of acid, and how does it deal with it? How does it maintain homeostasis? Well, first of all, it has a lactate or monocarboxylate transporter that takes one molecule of lactic acid, lactate, and one molecule of protons and exports one lactic acid molecule. It also, and now you can block this pathway with alpha-cyano 4-hydroxycinnamate, which is an inhibitor of this transporter. And later I found out that also a drug that Gil was very much involved in, Lonidamine, also is an inhibitor of this site. But we first worked only with the CHC. You also have a sodium-hydrogen exchanger here, which can pump out the protons and take in sodium ions. This is relatively inactive in our tumor, and it's probably inactive in quite a few, most of the tumors. This is really the preferred method of getting rid of acid. You can block this with cariparide if you have high levels of this transporter. And then there's the chloride bicarbonate antiport here that can extrude a chloride ion, and it takes in a bicarb, which can then neutralize some of the acids. But this is inactive under acidic conditions, so we're basically knocking it out as we're acidifying. So we don't have to worry about that. So our strategy was to trap the lactic acid inside the tumor cells by putting in the CHC as an inhibitor. And we found that this worked quite well. We could go down to a pH around 6.2, 6.3, but it didn't last very long. So you only got around 40 minutes overall, and that's not a very big window to apply cancer drugs. When we tried the Lonidamine, on the other hand, we found that we can get 2 to 3 hours of lowering of the pH down to 6.4. And there was also a de-energization, a very profound de-energization of the tumor, which made it even more susceptible to treatment. So Lonidamine has a lot of advantages, but it had one big disadvantage, and that is all the mice that we tried it on died overnight. So we eventually learned how to deal with this problem. But now let me back up and fill you in on this. We obtained a stimulus. This is thanks to President Obama. We were stimulated to look for a method, a safe method for administering the Lonidamine, and also to look at whether this effect was general to many other tissues that are involved in glycolytic metabolism, or was it specific for the tumor. And we were in fact able to demonstrate that it was quite specific for the tumors. And then we also wanted to examine the mechanism by which Lonidamine was acting. Now, back in 1983, Floridi reported that Lonidamine was an inhibitor of hexokinase 2. In the same year, he published a paper together with Al Leninger arguing that, in fact, it was inhibiting the electron transport of mitochondria. So there's a little bit of confusion over here. But then by 1995, the University of Tel Aviv, with Gil Navon and Ophir Kaplan and others, got to work on the problem, and they showed very clearly that the effect was not on hexokinase, but in fact it was inhibiting the monocarboxylate transporter. There was also a study by Mardor et al. That Jack Cohn was involved in, and these were really elegant studies involving phosphorus, C13, and proton NMR, but all were done on perfused MCF7 cells. In 1998, Ben Yosef at the University of Minnesota originally, and then I think they may have gone on to Michigan, did in vivo NMR studies using 9L glioma in the rat as a model. And again, they showed that there was acidification of the tumor with lonidamine, and they could also de-energize the tumor. And also they showed lonidamine is not very soluble. And whereas we were getting it into solution with DMSO, they actually had a nice method of using a pH 8.3 glycine buffer that got it into solution. And it's much easier to work with it under those conditions. So this is a story of a lot of Bens, Ben Horon, Ben Yosef. My name is Glickson, but my Hebraic form of that is Ben Mazel. So, okay, this is alpha-cyano 4'-hydroxycinnamate, which is Leninger's MCT inhibitor. And remember, this works very well, but it's only transient. This is lonidamine. It has some of the same properties, got a carboxy, an aromatic system, and still an unconjugated aromatic system beyond the conjugated aromatic system. So there's some similarities in structure, but I don't know if anybody's actually delineated the exact requirements for an MCT inhibitor. And this I've told you about. So here are our studies, our stimulated studies of the DB1 melanoma xenografts in nude mice. This is the baseline phosphorus 31 spectrum. I forgot to mention that we added 3-APP, 3-amino propylphosphonate, which doesn't get into the cells, but it's pH sensitive, so we can measure the extracellular pH. We have an intracellular pH meter from the inorganic phosphate, and of course we get all the energetics and the PMEs from here. And what we found was that after the addition of lonidamine, you found there occurred a large decrease in NTP. So there's energy that's being the energy production is being shut down. Inorganic phosphate shoots up. This thing shifts in the acid direction, and there's only a very small perturbation of the 3-APP by around 0.1 or 0.2 units. This summarizes all the results. So we find that the intracellular pH of the tumor, shown by these circles here, decreases from around 6.9 down to 6.4, and it just stays down there for at least 3 hours. The brain and the skeletal muscle show no change in intracellular pH. The liver shows a transient small drop and then returns back to normal. The extracellular pH is perturbed only for the tumor and very slightly. The lactic acid, which we measured with a Hadamard selective MQC sequence that we had developed, increases by about a factor of 3. And since the pH, the intracellular pH is dropping much more than the extracellular pH, we concluded this is mostly intracellular lactic acid being trapped in the tumor. And this shows you the NTP to PI ratio. It's monotonically decreasing in the tumor. It's doing nothing in the skeletal muscle or the brain. It shows a transient small dip in the liver and then again returns to its baseline level. So it is quite specific for the tumor, at least with respect to resting skeletal muscle, brain, and liver. This shows you the lactate, and you can see the large increase in lactic acid. So we had And the other thing is we left out our 26 millimolar glucose. That was the main culprit responsible for the mortality that we were seeing. So if we only gave lonidamine and counted on the intrinsic glycolytic metabolism of the tumor to generate lactic acid, none of the animals died. So we have a safe method to drop the pH significantly inside the tumor, and by the way, and reverse the pH gradient, which has all kinds of implications for delivery of other drugs, because the pH is normally more acidic outside than inside in a tumor, and now we've made it completely opposite. Well, we had solved those problems, and now we said, okay, let's see if we can find a cancer drug that we can activate. And there were reports that cisplatin was such a drug, and that cyclophosphamide and various other nitrogen mustards were. So these studies were conducted ex vivo, just on isolated cells. You don't normally give cyclophosphamide. It has to be activated in the liver. So we left that out, but we tried various other nitrogen mustards. And the most effective agent overall appeared to be Melphalan. And we had previous experience with having used Melphalan in treating melanoma in a different setting, so we were confident that that was a good choice. We went ahead and first studied the cisplatin, and we found that cisplatin, this is the baseline change in tumor volume as a function of time, and you can see that there's no difference between the control and the cisplatin. When you add lonidamine, there is some growth delay, but it's being driven by lonidamine. Lonidamine is actually more toxic than the cisplatin is, as you would expect, because it's lowering the energy of the tumor. But when we gave Melphalan, now we see a very big difference between the control saline-treated animals and the animals that were treated with Melphalan plus lonidamine. This is Melphalan alone, and this is lonidamine alone. So this is the lonidamine is producing a shift of about 2 to 3 weeks in the growth of the tumor, which, if you go through the calculations, is about 1 log order of cell kill with 1 treatment. This shows you that there is some toxicity to the whole body of the animal, in that there is a drop in weight with cisplatin by around 20% in this case, and with Melphalan by around 15%. But the animals recover their baseline weights within around 2 weeks after the Melphalan treatment. So this should be susceptible to multiple dosage therapy. This summarizes the results, and we get about 20-day growth delay in this experiment, only 10 days in this, but there was a lot of scatter in this data. We have to repeat that. But it averages out to about 15. I think when we repeat these experiments, we'll come up closer to the 20. And the doubling time also is about 6 days for this tumor. So we had an active agent with the lonidamine, and why should, if you increase the pH, say, by 1 unit, why should you get any more than 1 log? Well, we did get the 1 full log of cell kill, but there's multiple mechanisms that could contribute to the toxic effect of the Melphalan. Melphalan acts via an intramolecular SN2 reaction. The chloride is kicked out, and you form this active aziridinium ion, which is the nucleophile, that then adds to O6 or N7 of the guanine. One of the key scavengers, now, of course, this is going to increase in concentration with acid, because this nucleophile could be neutralized by hydroxide ions. So if you drop the pH, there's less hydroxide. Also, glutathione could deactivate this, and glutathione S-transferase activity is decreased with pH, as is the DNA repair via O6-alkyl transferase. So it's really a triple whammy effect that you have three different mechanisms that could contribute to the increased activity of melphalan and perhaps other nitrogen mustards. So lonidamine is routine now in terms of clinical translation. Lonidamine is routinely used in Europe and Canada, or has been at least, as an adjuvant to chemotherapy with various agents. And we were planning to go to Rome and conduct a study in Rome if we had to. The U.S. requires an IND. The FDA stopped sanctioning the use of lonidamine some time ago because of variable dosage effects and such. There was a recent clinical trial carried out by Threshold Pharmaceuticals. They were using this to treat benign prostate hyperplasia. They treated some 780-odd patients. They found 3 had an adverse effect on the liver with a drop of liver enzymes, nothing fatal, and they discontinued the study at that point. This is not a cancer, so if this had been a cancer, you would not have stopped the study with just 3, with some low level of toxicity like that. So it is possible to get an IND to do it in the States. It might be better, perhaps, to go abroad. In fact, 3 agents, other MCTs, are now being in clinical trial. They're produced by AstraZeneca, and they're in trial in India, I believe. No, I'm sorry, I'm confusing that. Lonidamine is also a male it's related to male contraceptives. Adjuvant is the hydrazone derivative that is used as an anti-spermatocytic agent, and it's in trial in India. But the clinical trial of the AstraZeneca, I believe, is in Europe and in the United States. So that might be an alternative to lonidamine. But we're thinking we should be able to go to clinical trial with lonidamine using an IND or going to a country that permits its use. Cisplatin turned out to be not activated by this approach, but melphalan appears to be a promising agent with the optimum dose and the optimum schedule yet to be determined. We need to do more studies of the toxicity to bone marrow, especially with melphalan, since it's known to have bone marrow toxicity. And in fact, that might be extremely interesting because the people working on multiple myeloma use that drug, and they're interested in seeing if lonidamine would have a beneficial effect. And we might be able to nudge the pH down below 6.4 a little bit by giving it a little bit of glucose, not large enough quantities to produce more toxicity. Okay, so studies that are planned or in progress quantitate the effects of lonidamine on glycolysis, identify the specific MCT isozymes. We should be able to find the specific isozyme in the cell membrane as well as the one in the mitochondrial membrane. We think that the MCT is acting not only to block export of lactic acid, but of transport of pyruvate from the cytosol into the mitochondria, and that's why you were seeing that energy drop. That's not an original idea. I think Gil and his group had come up with the same conclusion. And it should be possible to actually clone the MCTs and determine the exact fractional effect of lonidamine on each individual isozyme. Now, this is what I wanted to tell you about more than anything, is that we have been following more or less the prescription that Bob Shulman was proselytizing about in his speech, and that is quantitate. Determine exactly how much are you affecting glycolytic metabolism, how much are you affecting oxidative metabolism. Tony Mancuso has built a machine. I might say that I spent one summer here at the Weizmann Institute working with Hadassah Dagani on something similar to this, but there's a big difference. This is, in fact, an airtight system where you can control the introduction of how much oxygen is going in. We also have electrodes at the input and the output valve on this 20 millimeter NMR tube where cells that are adsorbed onto microcarrier beads are located, so we can measure the oxygen consumption rate directly in our cells. And then, of course, we can do the C13 NMR kinetics with this. So we wanted to analyze the kinetics and determine the actual flux through the various pathways. We did this in the heart, Langendorf perfused heart, back in 1995 with Teddy Chance. Ted's been retired and ill lately, and we got together with a very, very smart young Belarusian scientist who's with Camille Ugerbil. This is Alex Shestov. And Alex and Camille, they've been working on, and Pierre Gilly have been working on this Tumor method for analyzing fluxes. These are the data that we obtained. You can see we can clearly follow the glutamate C4, C2, and C3, as well as various other metabolites, and of course lactic acid. Then this was now analyzed where in the model we now include glycolysis, TCA cycle, the pyruvate carboxylase cycling pathways, amino acid metabolism, a variety of anaplerotic pathways, the pentose, the shunt, and that's about it. Now we're planning to add glycogen, although there's very little glycogen in these tumors. But for continuity and completeness, we will have glycogen in the model, and fatty acid metabolism will be added as well. This shows you all the fluxes that Alex has been able to determine using his modeling. This shows you the fit to the glutamate C4, C2, and C3. I think you'll agree that there's a good fit, and ditto for lactate. And these are the various fluxes that I showed you on that diagram. This is the important point. Everybody says tumors undergo aerobic glycolysis. Sure they do. But how much ATP do they get from it? And how much do they get from oxidative pathways? So what we found that under our conditions, which are not the natural, we have 26 millimolar glucose in these experiments. But we found that there was 54% of the ATP was being produced by the mitochondrial metabolism and 46% by glycolytic metabolism. Admittedly, a high level of glucose is going to suppress some of the oxidative metabolism via the Crabtree effect, and a high level of glucose will also favor a higher level of glycolytic metabolism via the Pasteur effect. But, so we're going to try to get this thing down to around 5 millimolar glucose to get it more realistic. But you can see that the tumor is not getting most of its energy by glycolysis. Even this tumor, which is relatively, this tumor is getting around 54, or say about 50, at least 50% via the glycolytic, via the oxidative pathway. And then we have to see just what does lonidamine do. Does it, according to Floridi, it should decrease glycolytic metabolism. We've already seen that it doesn't, but we can tell you by just how much. And we can also tell you by just how much the lonidamine will be decreasing mitochondrial metabolism. This is the validation of the model. These are the model predicted estimates of the CMR, the oxygen consumption rates. These are, I'm sorry, these are the modeled CMR02s, and these are the experimental. There's very good agreement. We also, in one of the experiments, were able to predict the glutamate pool size, and again, there's good agreement. So I think we have a valid model, and if anything, apart from being useful in this particular problem for studying the effects of lonidamine, this should be a very powerful tool for general studies of cancer. I won't tell you about all the other ones that are in progress. These are some of the additional people that were not co-authors that were contributing to this effort, and the first slide had the grant that was supporting it, which, by the way, scored a 3 percentile on the last review, so it's going to get another 5 years of funding. Thank you.
So congratulations on the grant. Have time for a short question? While you set up the presentation, Jared,
Yeah.
So there are two ways to change the acidity ratio between intra and extracellular. The other one is to give sodium bicarbonate to go the other way.
Right.
So do you see an advantage to one or the other?
Well, the bicarbonate is not going to do anything to intracellular pH, so it's going to, it will not potentiate the activity of a drug. Whereas it might increase the uptake of something like doxorubicin, which is a weak base. But this will have the effect of lowering the extracellular pH. So Bob Billy's idea behind bicarbonate was to cut down on tumor metastasis. And I think lonidamine will do the same thing. It should. We haven't studied it specifically, but at least from the production of lactate, it should be beneficial. But it will potentiate drug activity and hypothermia.